Hi, this is Dr. Ben Morrill. Welcome to episode 82 of Reptile Genetics Weekly. Once again, glad to have you here. Crazy day with all of the voting and stuff that's going on. It's nice that that's finally going to be over and not so dang many ads about that and people talking about that. I'm excited. I'm hopeful that we don't have to listen to a bunch more drama with that, but I guess we'll find out. But at least the voting will be over. But anyway, glad you're here to see what our race to 270 genetic tests will be and see where we're at on that. For sample updates, we have, as has been for quite a while, actually a good chunk of the year now, we have fast tests going out every day, pretty much every work day. And so your 1 to 2 business days turn around once we get your sample in the lab in most cases. And sex determination is 3 to 5 business days for ball pythons and colubrids. And then I finally do have some fairly concrete dates for the next panel run. So we should get the results early to mid next week, and then by the end of the week, so we're looking at about a week and a half from now, we should have the next panel results. So I know there's a bunch of people on that now that baby season's slowing down and moving out of that. We to be able to keep the costs cost effective, we have to batch them, and so now that the samples coming in are slowing down a little, and that makes it so that the panel tests will take a little bit longer. So we were doing them closer to like 3 to 5 weeks. It might be more like 4 to 6 weeks for the next little while. But you never know. We do have a special going on in November. So if we have a bunch of extra samples come in this month, maybe this next one will be a little faster. But yeah, we'll talk about that in just a second as well. Yeah, I think that's all I have right now. Kayla, how are you doing?
Hey Ben, I'm doing good. I had a very eventful weekend. Got tackled by a black vulture. I
Not many people can say that.
It's true. I'll have to pull up but message me if you want the video. It's pretty funny. Not a wild one. He was excited about his pumpkin, and I was too close to it, and he said, "No, it's my pumpkin," and did what black vultures do, which is tackle their people. Yeah, no, that was the thing. I cracked a rib by coughing, which was very an experience. I'm fine now.
I've had rib problems a couple times. That is no joke.
Excuse me, it is humbling. Yeah, no, it's literally just like I've been coughing for so long and stressing those same muscles. I was standing in the in the mammal room at work, and just a light cough, kind of like what I just did, and I felt and heard a crack near one of my sore spots, and I felt just a little dose of adrenaline. And I was like, did that really just happen? Did I really just do that? The first bone I've ever broken is done in the least cool way ever. Like, not like, breaking an ankle while doing like a kickflip or a sick kickflip or something, like, yeah, it's crazy. Saturday sucked, but it didn't, because, the day after is where you're really going to be in pain. Like, I was laughing on Friday about it. Like, yeah, this is my life. It's fine. It's hilarious, honestly. And then Saturday I wake up and I'm like, Jimmy, this is a lot less funny. Help me out of bed. So that was fun.
And anytime you laugh or cough or sneeze or anything, it just feels like someone's stabbing you.
Mm-hmm. I went to work the next day and they were like, I'm so sorry. Is there anything I can do? I'm like, no, it's fine. I'm on pain meds. It only hurts when I breathe, but that didn't stop me from, when we spotted this massive black racer outside the center, it didn't stop me from jumping out of my seat and chasing it down. This rib be darned, I will not be slowed down. And sadly, the racer got away. No pictures, no anything. But it was a chunk. It had to have been like 4 foot long, which is big for a racer. And so pretty and very spicy.
Suckers. Oh yeah.
They are. I got like this far away from it, and it zipped out. And we were like, my coworkers were helping me. They were like, I don't know how to grab a wild snake, but I'll help you like poke a stick to get it out from under the shed. And zoomed past me and it was gone. Didn't see it again, but so pretty. It was my lifer too, so at least I got my lifer black racer. So That was good. I don't think there was anything else too crazy this weekend. Those were the main ones though. So it's so I'm feeling a lot better than I did Saturday, and we're moving right along.
Good.
So let's get some business stuff out of the way. Right now is an amazing time to get stuff off of Morph Market, stuff specifically being shed tests and morph tests and sex determination tests, because Morph Market is giving you 25% off all shed tests all November long. So definitely want to take advantage of that. It's really nice of them to, partner up with us and to do this. So you can buy them now and then use them later on. So this is like the perfect time to buy them before you get next year's clutches out.
Yes. Yeah. They will never go bad. So you can pay for them now and get a really good discount and use them in 4 months or 6 months or in a year or whatever. And
yeah,
so it's a great way that, like we've talked about before, this is something to help us kind of pay the bills through the slower months. And as a thank you to those of you that are willing to prepay, we'll happily give you a better deal. So jump on it while you can.
Yeah, buddy. And of course, we have our bit about bulk discounts in general. We've played these before, but if you really want to see like how steep of a discount you can get for buying shed tests in bulk, you can get a quote from us by emailing Sean at raregeninc@gmail. It's like, especially with colubrid sex determination, if you're expecting, several clutches of babies like corn snakes or hogs next year, you can get them down to as low as $15 per test. And that will save you the popping and wriggling and tiny biting and musk flinging. It can save you the stress.
Yeah.
So definitely something to look into. And did I miss anything on that, Ben?
And I would just say it's even better to send that quote for a bulk order to Sean during November, because you already know you're at least going to get 25% off. And if you have enough numbers coming, he'll likely be able to do even better than that. So this is the time to do it. This is a great time to do it.
Definitely. All right. So that's a really nice thing. And we're actually going to be at the St. Louis NARBC show. St. Louis. How did the locals say it? Someone help. But we're going to be there. So at the St. Charles Convention Center, Sean will be there. So you can catch up with him. Definitely hand him sheds, whether that is, tests that you've ordered, or if you have samples that you want to help us out with for future research runs. We'll be taking them there. And also Sean's...
He'll do a better job than I did. Those of you that went to shows, I was the only one there. Sorry, I didn't do a very good job of getting your tests run quickly. Sean will do a better job of that, because he'll take it back. The Texas lab is the one we do stuff fast. I get stuff done here, but he gets it done fast there. So
oh yeah, Sean's got the
Yes, that will be good. The other thing is he will have at least shirts. I don't know if he'll have anything else, but he'll at least have shirts there, and they're, specifically for 2024. They'll have the morphs that came out, over the year previous. And so next year, he was kind of joking, we might actually have to do 2025 A shirt and a 2025 B shirt, because we'll likely have a whole bunch more next year.
Oh man, I haven't even seen the design, so I'm excited to see how those look.
Yeah, it's pretty cool. It's similar to the one we had the first year, but it's got more morphs on it because we have more tests come out. So,
ooh, exciting. Well, awesome. We'll sound off in the comments if you're going to be at the St. Louis show this weekend.
Yes.
So the race to 227 genetic tests. So Ben, what is, why did I say 207? I meant 270 genetic tests. Whatever I said, words are hard. What is your, what's the thought process between 270 tests?
Oh, just because of the voting tonight, you hear all of the stuff about, who's going to get to 270. So this is our 270.
Oh, got you. I didn't catch that. That's cute. I like that. Oh, yes. Awesome.
So that's why we're hopeful we can get into the couple hundred tests. 270 might take a little while, but I think once we get things flowing in other species, some of the tests will come a lot faster. So we're getting better at it, faster at it. We'll likely kind of like the last batch of new tests, we had 10 at a time. That'll probably be more likely what will happen when we do release stuff. It'll be like 5 to 10 or a dozen or something like that at a time. With Monsoon, since we've put so much work into it and worked with other people, that's one that we're very close. And we talked a little bit about in a show recently and did a post about it. That one is 98% correct. That one is really, really good, but it's not perfect. We know that it makes some bad calls. We had both one that I ran and one that Charlie ran that were clearly not visual monsoons and shouldn't have been, but they tested homozygous. So we know that test is not perfect. But that was 2. There's one other one that tested incorrectly. So that was 3 out of like 215 or 220. So we're all still working on getting that, just that last little bit. We know for sure we're in the right region now, so we got to find the right mutation and get that test done. But that's one that we might release on its own because so many people have been waiting for that for so long. But other than that, we'll probably release 5 to 10 or 12 at a time, something like that for the others.
Awesome. So how is Monarch looking?
Monarch is still stuck at the kind of 80 to 90% correct. We do have a test that's right a good chunk of the time, but I haven't been able to improve that in a while. I have focused more on Monsoon than Monarch, but I will have a whole bunch more data coming here soon that Monarch will be a part of. So yeah, awesome. we're gonna sometime soon. We're gonna do an episode where we really go in depth about that process, what it's like when we say, oh, we think we're really close, and then we end up saying, never mind, it's going to be a while, or we say where we think we're close like 5 or 6 times and keep failing. We're going to kind of give you guys a detailed idea of that process, what it's like, and give you an idea so you don't think that we just suck at our jobs or we're crazy. We're liars or whatever. We really are working hard and trying our best. This is just how this kind of test development goes. It's an iterative process, and sometimes you're on a path that looks really good, and then you find out it's a dead end, and you got to go back to the very beginning again. And sometimes you got to do that 70 times. Well, not 70. We haven't done that yet, but we're with Monsoon and Monarch. We're probably on like 5 or 6, something like that. So of rounds and each round costs money and time and focus. And anyway, so yeah, we'll do a whole episode where we'll go into some specific numbers, what it's like, and how often we get new tests and then find out there's more than one mutation, like with yellow belly, and we know for sure with special now and stuff like that. And so anyway, there's a lot of cool detail we'll go into in that episode in more detail.
I'm looking forward to it.
Yeah.
So where are we sitting with confusion?
So confusion, we know we're in the right area because we do have an acid test that works well, but the confusion test is also in that like 70 to 85. I think that one's over 80%. The test that I have for that, I actually have 2 different tests for confusion, but both of them are only in that like 80% range. So
okay,
that one also, like Monsoon or Monarch, I'll have more data in that big chunk of data I have coming. So
okay. And what about red stripe? So we have that test, but so there's an update going on there. What's the situation?
Yeah, so with red stripe, what we think we're seeing, the feedback we're getting, and thanks once again to all of you for using testing and sending in sheds. And then when we give you feedback back, re-feedback, I don't know what you call that, but like we say, hey, can you send me the sire and the dam, or blah blah blah, or help us kind of trace lineages to figure out where. So the thing that we see is every once in a while we'll have a red stripe like in a panel or from a breeding, like the breeders like, hey, from these babies, this one has to be a red stripe, but it's testing negative. So there's just some times where it seems very clear that that animal, and this is including some that Justin Kobelka has and some other people that, have seen a lot of red stripes, so like, this one has to be, and we've had Justin send us several that he knows for sure are, and then a few of those have popped negative. And so we've kind of figured out it's likely there's a second mutation that also causes that red stripe look, which we know now is not an uncommon thing. That happens a lot. It happened with yellow belly and with ultramel and with for sure with special, and many more that we'll talk about some others here in a second too. We'll be doing updates on. But yeah, it seems to be a fairly common thing. And so the interesting thing with red stripe, and we just talked about confusion, so far in breedings where people have bred a confusion red stripe or acid red stripe to something, it's acting like a super. And so we're pretty confident. And what we're seeing in the tests that we have, they're fairly close to each other. So it may be like banana and the sex determining region and ball pythons where 96% of the time they'll travel together, or it may be closer to 100%, or it might be at like 85%. We don't know, but they seem to be close enough that they travel together. And I guess they could even be in the same gene, like yellow belly and asphalt and gravel and all of them, but we'll see. We don't know for sure yet, but they are close enough. There seems to be some, it's called linkage disequilibrium. There seems to be some linkage between red stripe and then confusion and acid. And so we're playing with that. And another one that I'll have more data on in the next week or two. And, it could be that data comes through. I'm like, oh, this has got to be it. And it works well. And, in a couple of weeks we have more information, or it could be that it's not helpful at all and we got to sequence more. So, but yeah, we're working on those and making progress.
That'll be exciting to see. So what about black pastel? I know there's been some stuff going on there too.
Yeah, so with black pastel, we knew that cinnamon looks similar. Sometimes people aren't totally sure whether they have a cinnamon or a black pastel, and our black pastel test is definitely negative for any cinnamons. I've run several from different people. It's always negative. So cinnamon is definitely a different mutation, and it does look a little different. So that's not surprising. But we did come out with a Barnhart black pastel test. And we still have some animals that, especially in Canada, the feedback we got from Canada, their main line of black pastel up there tests negative for our black pastel test. And then for us, we have some people, they'll send the picture like, hey, yeah, this definitely looks like it is a black pastel, but it tests negative for our black pastel and for Barnhart black pastel. So there seems to be at least 3 different black pastels. And then you have cinnamon on top of that. So that's 4 different ones that look very similar, but there is at least 4 different mutations. And we could find out there's even more. Maybe we find that 3rd black pastel line, and then we still have some that come back every once in a while negative. It could be another mutation, So anyway, it's something that we can't make that progress without you all helping us. And so we're very thankful for people that let us know, hey, this test doesn't make sense. That's one of the biggest reasons why the panels are such a benefit, because sometimes people send something in that they know is a black pastel clown, and it tests, and they're testing it for posset hypo and posset, this and that and the other. But it comes up negative and they say, hey, here's the picture. It has to be a black pastel. We're so thankful that people do that, because that's what helps us key in and know, all right, there's more work to do on black pastel. So any of you that have had a black pastel or red stripe we just talked about, that you're pretty confident it's there, but it tested negative, just know we are working on updating those. And each month that goes by, we'll know a little more. And as soon as we know something for sure, we will definitely let you all know.
Man, so much going on, and just it's so funny where we think we have something locked down, and then these snakes genetics decide to throw us a curveball. Even if you're not the one hatching the eggs, it's going to happen. So
yeah.
All right. So what about special? Adding on to the same line of updating these curveballs.
Yeah, so this is another one that we'll be updating. We have enough from Twin City Reptiles for their TCR line of special that definitely does test negative for our special test, which is the one that Dr. Sadell found, and I'm pretty certain anyone doing tests is using that same test. But the TCRs definitely test negative. It's a different mutation. Obviously looks similar, but a little different in some of the combos. And then there's another line called the NOCO line, or Northern Colorado reptile, Northern Colorado or NOCO reptiles had that line. What we don't know is if it's the same as TCR or if it's a third one. We just know that both of those lines, when we've gotten those tests negative for the current special test, so they're not like really prevalent in our at least U.S. population. These two other lines, but the few individuals we have definitely do test negative, and so we are working to sequence those individuals, multiple individuals for both of those lines, TCR and NOCO, to be able to update that and be able to say, hey, here's a second line, a second test that covers both NOCO and TCR, or maybe we'll have to say, hey, here's special, here's NOCO special, and here's TCR special. We'll find out over the next month or two, probably.
Yeah, and special is part of the blue-eyed Lucy complex, right? Yeah. Okay, that's what I thought. That complex, these complexes keep having more and more things added onto them, it seems.
Yeah, it can get complicated.
So once again, going into the yellow belly complex, we have Asphalt.
Yes. So we've had a couple of customers, at least, with very frustrating Asphalt results where we've tested and retested and, asked for a sire down, all this kind of stuff. And it just really does test negative, even though the animal should be an Asphalt. I've had some people send in, I think I've had 3 different reptile creation line Asphalt or RK Asphalt. Thank you for those of you that have sent those in. And in every case, when they've sent those RK line Asphalts, they test negative by the test that I use. They use it in Canada, they use it in Europe. Like, it's the test that Dr. Sadell published. But there seems to be at least one other line of Asphalt also. And once again, it's not really prevalent in the United States. So the chances that if you have Asphalts that yours came from that line is not very high. But it is something to keep in mind that it's a possibility, and it's something we want to be able to make sure we can test for our customers. So we'll hopefully have that as well in the next month or two. We'll be doing some sequencing on those 3 individuals that I have, and hopefully we can find it, since it should be, near the other yellow belly complex animals. We know where to be looking, and so hopefully we'll nail that down.
Very cool. It's kind of like the Ultramel B of the Asphalt gene, I guess.
Yeah. Yep.
Yeah. All right. So high intensity OD. How is the test for that going?
Yeah, so I got to talk with Chris Hernan and with Wes of Muscles and Geeks Reptiles and quite a bit at Tinley about this. And it seems like what's happening is the high OD line does test the same as OD, but clearly there's something different that amps those animals up. So what we are doing, we're collecting even more. Both Chris and Wes gave us a whole bunch of samples, but then some other people that have heard us talk about it on here have also sent in, and even if you have only an animal or two, if you want to send it in, let us know, whether it's a high OD or super high OD or what the pairing was or who it came from, or if you produced it, just as much information as you can. But we're continuously sequencing more and more of these. So it's kind of a more complicated thing to find. I'm assuming that what we'll eventually find is there's a secondary mutation close by in the ones that are super OD, high ODs. I mean, those high ODs have a secondary mutation that kind of amps that up, but we don't know for sure. So we're going to do, it's going to take more sequencing than finding OD originally. We're going to have to do even more sequencing and hopefully be able to find that secondary mutation. But we may not ever be able to, or it might be really easy and we'll find it in a month or two. We'll see.
Well, hopefully we can, because they do look really nice. I do like a good IOD.
Yeah.
And so lace and white lace.
Yeah, so we got a whole bunch of samples. Justin Kabelka really focused and pulled a whole bunch. And he said he specifically pulled ones that he is confident are white lace or confident are lace. Animals like in the lace project, one of the biggest difficulties is that sometimes when you hatch out babies where they're not coming from a white lace, you don't know for sure if it's a lace breeding. You don't know for sure which ones have lace and which ones don't. And as they kind of right when they hatch out versus after 2 or 3 sheds versus after a year or two, it can be more or less difficult to kind of make those calls. So we do have a whole bunch going to sequencing. So it's going to take a little while for us to get all of that data back. So it's probably at least a couple of months out before we'll get a big batch of data for lace. But it's very likely that when we do get that we'll be able to nail it down, because we've had a concerted effort, Justin especially, but then some other people have as well. And once again, thank you all that are sending sheds in. You are the ones that make this possible.
Yes, they are. So Mandarin, how are we looking with that?
The Mandarin is right there with lace. Justin Kabelka collected a whole bunch of them that he knows for certain whether they're Mandarin or Super Mandarin. So that's really good, really important to have samples that we have high confidence have been identified correctly. Mandarin is in the same boat. Sometimes it's a little difficult to know for sure what you have. So that's also going to be in the same boat. With lace, it's going to be at least a couple months, but we are working on it, and we've got a good batch of samples coming through, and so we're excited about that.
Awesome. So Zebra
could pop loose any day. I don't have like a 70 or 80% test yet, but I do have a decent location, and that's another one in the next week or two I'll get more data on, and it could pop loose, or it could be one that I got to work on more. So we'll see on that one, but it's hopefully coming soon.
Hope so. I've got a soft spot for zebras. Like, I was looking for photos to pull for, so we could give examples, and oh my gosh, the combos it makes are so pretty.
Yeah.
Like, this snake is just like, oh, amazing. Gold star Adam Chesla reptiles. So GCR and MJ Axanthic, which
so this one is in the same boat as Monsoon. This one, we have a test that is correct 98% of the freaking time. Unfortunately, in this case, there were 2 or 3 samples that, from everything that I know and that the breeders knew, there was no axanthic lineage. They were ones I picked as known negatives to run as part of the validation. So when I'm validating a new test, I have to run homozygous animals. So in this case, actual visual GCR and or MJ axanthics. Then I also run known hets, and then I also run known negatives, because I need to make sure the test works on all 3 of those. I need to make sure if someone sends a shed in from a posset clutch that's negative, that the test will say negative, and if it is an actual het, that the test will say het. So those negatives are just as important. People probably think a lot about, how many homozygous and heterozygous ones did you run, but it's actually just as important, if not more important, to make sure that the negatives are called negative. And so the only problem that I've seen so far with GCR and MJ is where 2 or 3 animals that were from lineages that there's no known axanthic anything and shouldn't have been any, but they tested as hets. So that's concerning.
That is.
So it's really dang close. Obviously, again, that was another close to 200, and we only had, a couple, I can't remember for sure. It was either 2 or 3 that were problems. So there's like about 200 that were 100% correct. There's just a couple that were wrong. So anyway, that one's super close, and hopefully we have it. I will have more data specifically on GCR and MJ in the next week or 2. So there's a chance, or we may have to go back to the drawing board, but at least we know we're in a pretty specific region. We know that has to be the correct region.
All right. So Stranger.
Stranger's one that also could pop loose. I'll have more data in the next week or two. We have a not as specific a region as Monsoon and GCR and MJ, but we do have a little bit broader region where we know it has to be in there. So it's looking good, but you know, it could pop loose in the next week or two, or it could take another round of sequencing. But it's one that we have made some significant progress on.
Awesome. Now Mahogany. How's that looking?
That one's right in the same boat as Stranger. That one could pop loose in the next week or two. I'll have more data on that one too. So lots of good stuff coming. Yeah, lots of good stuff coming in the next week or two. And then there's going to be kind of another big batch in about a month or two when I get another big run of sequencing new stuff coming through. But Mahogany and Stranger are both in the 1 to 2 week range. I have a chance of hitting them.
All right. Because you do a research run whenever you also do the panel runs, right?
At least. Sometimes we do. In addition to that, yeah, we're doing lots of these are all thousands of dollars runs. There's lots. We definitely are putting a lot, but right back into everything we're doing to make more tests. Our goal may not necessarily literally be the 270 like in the next year, but 150, that's I think something that we could reach by the end of 2025. So we're close to 50 now, but I think we'll be able to find them a lot faster and in other species and stuff like that. So it's going to be a fun 2025 is going to be a lot different ride than 2024 and 2023 have been.
I'm excited. So how about caramel albino? Caramel, however you pronounce it.
So I've had many people request a test for caramel, especially people that have ultramels, and they're concerned that there's some caramel albino mixed in. For those of you that aren't familiar, the caramels are famous for having some kinking problems, and then also the females are not able to reproduce.
Oh wow!
So it's a significant issue. They're beautiful, and it's really unfortunate. One of the first year there were ball python combos made. One was the bumblebee, and one was a caramel albino with hypo, and they called it a caramel glow. But it's been a part of ball python breeding for a long time, but people have bred away from it. But every once in a while, someone working with ultramels will pop out a caramel and have some problems. And so lots of breeders have asked me to make a caramel test. And then I say, okay, great. Give me some known caramels as samples to be able to develop the test. And for a long time, nobody ever actually gave me anything. But I did finally get a decent amount. I think it's like about a dozen. So I don't know for sure if we'll get it. And this is kind of in the more 1 to 2 month range when we get more big batch of data from sequencing in a month or two. That we hopefully can, if we get lucky, we can find that. And then that would be a test for, people that have ultramel or other animals that come from a background where there were some caramels in that line, that you could use that test to essentially breed that out of your population. And so we're working on that. Very, very thankful to those of you. When that test does come, you all are definitely going to hear who helped make it happen, as long as they're cool with me letting people know. Some people that have caramels don't want people to know they have caramels because they don't want people to be worried that they're going to end up getting something. So, but anyway, we're thankful that we have some. And any of you listening, if you have some caramels or know somebody that has some known caramels, we could definitely use more sheds to make that test.
There you go. All right. Phone. And so next, let's talk about non-ball python species. So our bloods, our retics, our berms. Oh my.
Yes. Yeah. So in 2025, we expect to be able to develop tests in these other python species, at least these three. I do have some carpet python, especially like axanthic carpets. We're doing a lot of axanthic and other species. Obviously, we've done it in ball python, so we have it coming in some other species too. other one that I've got some black-headed python, axanthic black-headed pythons. I think I might even have some albino and olive pythons. So some of the even less common python species, I've got samples going. But bloods, retics, and berms, we should have multiple tests in 2025.
Oh, that's awesome. Any sex determination tests for them as well?
Yeah, we're working on that also. That's been more complicated with all of colubrids. We were able to find tests that were fairly general, and
Mhm.
it wasn't too hard to figure out how to do the vast majority. We obviously haven't tested every colubrid known. When someone sends a species, that's why we always ask what species it is.
Yeah.
They send one in we haven't tested, we let them know, hey, we haven't done this one before. So we'd love to, if you have a known male and female to send as well, we'll run those and make sure our tests work on that. But yeah, we definitely would like to develop those in pythons. The colubrids, it was easy to find general ones that covered several species, several genera. But with pythons, the ball python one doesn't work on any other python species. And we've found out in Morelia, especially, that's the green tree pythons and the carpet pythons, their
Yep.
sex chromosomes are very different from all other python species, all other genera outside of Morelia. So the sex chromosomes in pythons seem to be much more variable than what we saw in colubrids. So it's going to take more work for each species of python than we had to do for all the colubrids.
Fair enough. I think evolutionarily speaking, I know that I'm pretty sure pythons and boas are a more basal lineage of snakes. And so I wonder if the fact that their sex chromosomes are so wacky has something to do with the fact that they're just older than, say, some, I don't know, some colubrids and vipers and what have you. That's just me spitballing a random thought, though, so it may have nothing to do with it. I know that there's a paper about Morelia pythons coming out as well at some point, but
We sure hope so. They're supposed to be.
fingers crossed, because I want to read that too.
Yeah.
So, side tangent, onward. Let's go to corns, hogs, boas, indigos, and maybe even some lizards.
Yes, yeah. So in 2025, we should be able to have at least some tests in all of these groups as well, and hopefully at least a couple of lizard species. We've
Oh,
always
nice.
been interested in blue tongues and monitors and heloderma, and some of the really common ones like crested geckos, and there's many things we would love to be working on, but we've got to get through some more ball python tests and kind of break into a few new species. But I think once we get all of that smoothly rolling, it will make it even easier to pick up other new species and get a few more developed. So that's why we're thinking that 2025 we should have a lot more tests coming.
Awesome. And for those of you who are sitting back like, come on, it's always the ball pythons. Ball pythons are help paying the bills for these other tests, because it's really expensive to sequence these and to run these different things. So that's the main reason why and also there's a huge demand for it. So
Yeah.
keeping the business solvent, while also solvent is the correct word, right? Yeah. Keeping the business solvent in addition to going down these routes of species that we also really, really want to see, who maybe aren't on every single table at an expo.
Yeah. Being a reptile keeper and breeder for over 20 years, that's what I want the most, is to develop tests that help lots of other breeders and keepers. And so the ball python one, there's so much interest and money in that helps us to be able to learn. First of all, some of it's learning. We're learning how to find new mutations faster and better and more efficiently, and then also to know where to look and things like that. So not only is the ball python work helping to pay the bills to be able to do other species, but it's also helping us learn how to do it so we can do it more efficiently. So when tests come out in other species, they're going to be less expensive. We're not going to go back to charging $65 a test. More than likely, when these new species, when tests come out for them, they can be more in line with, after we've dropped the ball python prices a time or two or whatever it ends up being, those will be able to come in at a similar level and not have to pay as much as we did early on. And that is a big part of it. Is it's great to make things cost effective and cheap and all that, but if you make it too cheap and your business goes out, then that doesn't help your customer at all.
Yeah.
If we go out of business, then we don't have anything to help you with. And just hope that someone else does an okay job, but we would not be able to help. So yeah, that's a big part of it, is making sure people understand what we're doing and how it can help them. And then also to have enough money coming through that we can do other side projects and things and get them going. And, if at the end of 2025, if we have as many tests in other species as we do in ball pythons, we may only have 3 or 4 or 5 in one species and 7 or 8 in another species. But if we can get, similar numbers, might be more species, many more species, but a similar number, then that helps our volume. That would essentially double our volume if things were linear. And then that makes it so we can do everything cheaper, whether it's sex determination or another new morph test. If we can double our volume coming through by the end of 2025, because we have twice as many tests and several new species, then that makes it so we can get those tests done for you at a cheaper price. And that's what we're hoping for.
Yeah. All right. So this week's shed donor shout out is McClure Hatchery. They have sent in sheds that have helped us develop new tests, and we're very grateful. So we wanted to make sure that you guys got a chance to check them out. They have really nice photos on their Instagram of eggs. You've got them candling some little growing embryos there. Lots of pretty
Beautiful
ball pythons. And they're also on TikTok. So if you're on there and you like to follow people who do snake stuff, they're definitely someone to check out. And that's McClure Hatchery.
And beautiful animals. And thank you once again for sending in sheds. That's like we've said many, many times on the show, even this show today. That's literally why we can do what we're doing, is because of people sending in sheds and letting us know what it is. And then we put that all together and figure out new tests.
Definitely. And on their bio there, they said that it looks like they're actually one of our neighbors. They're not too far away in North Carolina, and it looks like Jaguar is one of their main projects. So if you're in the Jaguar, they'll definitely be somebody to follow.
Yes. Yeah. They did send in some Jaguar sheds.
Yeah. Well, that makes sense. Yep. Awesome. All right. So I think that's everything we got for today. So if you like this, general roundup of where we are with the more popular morphs, definitely let us know. Oh, weird. The formatting on the Scout all wacky. I don't know how, but whatever. It's fine.
I guess you had to upload it a different way this time, right?
That's true. Well, that's okay. It's, if it wants to be creative, who am I to stop it? So everybody make sure you like, comment, subscribe. It tells the algorithm that we're cool and that, other people should watch our stuff, especially if they're interested in snakes and genetic testing. So yeah, thank you guys. And Ben, we got anything before we call it a day?
No, that's it. Thanks again for watching, and we will be back again next week.
All right. Thanks, guys. Don't forget to go out and vote if you're in the US. Catch you later. Outro in 3, 2, 1.